In this lecture, I will compare B cell receptor and T cell receptor, and these are the 14 points. The first is components. The B cell receptor is composed of IgM and IgD antibody, that is membrane bound in conditions, membrane bound. In case of T cell receptor, it is alpha and beta chain, or it can be gamma and delta chain. In our body, in adult body, 99.5% T cells are alpha beta type, and only very few 0.5% of cells are gamma delta type, associated molecule, associated signaling molecules. In case of B cell receptor, this signaling molecule is called Ig alpha and Ig beta. In case of this IgM and IgD antibody, the cytoplasmic region of the antibody contain 3 amino acid only. So the associated molecule actually perform the signaling process like Ig alpha and Ig beta. For T cell receptor, the associated signaling molecule is called CD3 and zeta. So these two molecules actually perform associated signaling molecule, perform the signaling process. The next is uniqueness. In my previous class, I have mentioned basic concepts of B cell receptor and basic concepts of T cell receptor. I have mentioned each B cell and each T cells express unique B cell and T cell receptor. That means if I have 10 million B cells, then I have 10 million unique B cell receptor that can recognize 10 million different antigen. The same is true for T cell receptor. If I have 10 million T cell, then I have 10 million T cell receptor that can bind 10 million different antigen. So they are unique. So for the B cell receptor, uniqueness is true, and for T cell receptor, uniqueness is true. VDJ recombinations. So as I have mentioned that if I have 10 million B cells or T cells, I have 10 million B cell receptor or T cell receptor. That means diverse number of B cell receptor and T cell receptor. Where from this diversity come? A process called VDJ recombinations or somatic recombinations occur in B cells. For the B cell receptor production, diversity of B cells receptor and diverse set of T cell receptor also produced by this VDJ recombinations. So both actually this B cell receptor and T cell receptor perform this process. The next is allelic exclusions. What happens? I have also explained that B cells and T cells both go through allelic exclusion. It means if a B cell starts to produce antibody from IgM or IgD from father chromosome, it excludes the mother chromosome to produce the same antibody in a cell, particular cell. Let's assume it is a B1 cell and it is a B2 cells. There are many cells, but I am just comparing these two cells. If this B1 cell is producing antibody from the father, it will not produce antibody from the mother chromosome. It excludes. If B2 cell is producing its antibody from mother, it will not express or produce antibody from the father chromosome. It will exclude. The same is true for the T cell, that is T1 and T2. Both these cells, like B cell receptor, go through allelic exclusion. In case of T cell receptor that contains alpha and beta chain, the beta chain maintain allelic exclusion very well, but the alpha chain in case of allelic exclusion is somehow leaky. So alpha chain is leaky. What does it mean? It has been found that in an adult human body, approximately 30% of T cells are present that express alpha chain from both the chromosomes. The next is endocytosis. The B cell receptor, like if this is a B cell, membrane bound antibodies, it may be IgM, IgD. When it binds with the antigen or pathogen, it performs one thing it called receptor-mediated endocytosis. Actually, by this method, receptor-mediated endocytosis, this B cell actually engulf or enter this antigen along with the antibody and chop or process the antigen and represent this antigen or processed antigen by the MHC2 molecule. But T cell receptor do not perform this type of function. So this is not performed by the T cell receptor. Okay, antigen type. What type of antigen the B cell receptor can bind? It has been seen all type of antigen, all type, like protein, lipid, polysaccharide. Any type of antigen can be bound by the B cell receptor, but in case of T cell receptor, only peptide or protein in nature antigen can bind with the T cell receptor. Okay. The next is bind soluble antigen. The B cell receptor can bind with soluble antigen, not membrane bound or any bound antigen. It can bind with soluble antigen. Soluble antigen means it is moving. This antigen is moving through blood, soluble. Now it can bind with the antibody, but T cell receptor cannot bind with that soluble receptor. So for the B cell receptor, binding soluble antigen is true, but not for the T cell receptor. Now, type of epitope bind. Epitope can be of two types. One is conformational and linear. Conformational means, at first I want to mention what is epitope. Let's assume this is an antigen. The whole antigen, this total antigen is not responsible for the binding with the B cell receptor, T cell receptor. This antigen have some portion that is actually responsible for the binding with the B cell receptor and T cell receptor, that is called epitope. A portion of the antigen that is responsible for the binding with the antigen receptors. So antibody binds in this region. So they bind in this region, not the whole antigen. So epitope can be of two types, conformational and linear. Conformational means if the epitope is actually present as a alpha helix or beta sheet, then it is called conformational. And if the epitope is actually processed in a professional antigen presenting cell, is processed or denatured so that it becomes linear, this B cell receptor can bind type of epitope conformation and linear, but T cell receptor can only bind linear epitope. Required help to bind antigen. Is this B cell receptor required any help to bind this antigen? The answer is no. There is no need or no help required for the antigen binding. But in case of T cell receptor, yes, there is requirement of a help from the MHC molecule, major histocompatibility complex. It can be MHC1 or 2 depending on the type of T cell. If the T cell is cytotoxic T cell, it binds with the MHC1, and if it is helper cell, it will actually bind with the MHC molecule MHC2 type. So it requires a help MHC molecule. The 11th is soluble form. The B cell receptor, that means IgM and IgD, can be in soluble form. When the B cell actually activated, it becomes plasma cell. Now it can secrete antibody. So B cell receptor or antibody can be secreted or soluble in form, or it can be secreted form. But the T cell receptor cannot be secreted in form. It cannot be secreted. Okay. Class switching. In case of B cell receptor, class switching occur. Class switching means at first the B cell actually expressing IgM and IgD antibody. These are two isotypes, but there are three other isotypes like IgG, IgA, IgE. So these are all 5 isotypes. But B cell can sense what type of antigen or pathogen it is, and it can secrete or produce that type of antibody that will actually against the pathogen. So IgG, IgA or IgE, it can change or can switch the class or isotype. It is also known as isotype. So it can change or switch the isotype of the antibody. Yes, but in case of T cell receptor, they cannot change the isotype. It means if a T cell is expressing alpha beta chain, it cannot switch into gamma delta, or the gamma delta T cell cannot switch between the alpha beta chain. The next is receptor editing. During the development of B cells, some B cells are produced, or during the VDJ recombination, some B cells receptors are produced that can recognize self-antigen. They are called self-reactive. 3 things can occur for these cells. One is deletion or killing of the cells. Okay, this is called clonal deletion. Another is it can become non-functional, non-responsive, called anergy. Or it can have, the B cell can have its second chance to edit its receptor or B cell receptor so that it cannot bind with the self-antigen. So this is called receptor editing. The B cells go through a receptor, one chance of B cell receptor editing by which it cannot bind with the same antigen or self-antigen. So they go through receptor editing process. But in case of T cell receptor, if during the VDJ recombination, if some T cell receptor are produced that can bind with the self-antigen, they don't have the second chance for the receptor editing. They actually simply die. That is called negative selections. They also go through this process. So, but receptor editing do not perform by the T cell receptor. They do not get the chance. Affinity maturations. What is affinity maturations? After second time infections, the antibody produced by the same B cells can bind with the same antigen but with high affinity. Affinity gets increased. So this process occurs in case of B cells. So affinity maturation occurs. The B cells can produce antibody, same specificity, antigen specificity remains same. It can bind with the same antigen but now with higher affinity. This is called affinity maturations. But in case of T cell receptor, this process also not occur.